cloudy evening. It's just so perfect out there. It's really lovely to be here with you all and to talk about submerged aquatic plants, which I've been thinking about ever since actually my graduate work. But that's not where I started. So I'm a plant ecologist now, but this is not where I started. Dennis just said that I did my undergraduate at Oregon State, and this was my first foray into research, where I worked with feral horses in the deserts of Nevada and California. So you can't get any further away from aquatic plants than what I worked with there. But then I got interested in ecological research and went to graduate school where I got a research project to work with trumpeter swans. This individual actually comes from the Rocky Mountain population. Populations in the United States. There is the Pacific population, the Rocky Mountain population, and then the interior population, which can migrate down to the Great Lakes, and sometimes you actually see some individuals actually showing up here at CERC. And so I actually found this neat article that actually just came out recently, showing that you actually have some trumpeter swans here at CERC. So this was actually really exciting for me to see, and then I thought I have to show you my trumpeter swan picture. Because then the trumpeter swan then leads into submerged aquatic plants. So in order to understand trumpeter swans and whether we can translocate them from an area in Yellowstone down to Utah, which is something that I was working on, we need to really understand their food resources, whether they have enough and what they eat. What they were eating in Utah, and what we have here in the Chesapeake Bay as well, is what is called sago pondweed, Stauchinia pectinata. And it can grow really, really densely, and it produces these tubers which the trumpeter swan feeds on in the winter. So trumpeter swans and submerged aquatic plants really go hand in hand. In order to understand trumpeter swans, we need to understand the food resources, as well as in order to understand submerged aquatic plants, you actually also have to understand how they are dispersed, and waterfowl could be a dispersal agent for submerged aquatic plants. So submerged aquatic plants obviously then are important for waterfowl and for, just generally animals. But submerged aquatic plants are also very important nursery habitats for crabs and other shellfish. And then also submerged aquatic plants are important for sediment trapping, nutrient absorption, and shoreline protection. When water flows through a submerged aquatic plant bed, the water energy dissipates, and so therefore that allows sediments to settle. Also, submerged aquatic plants pick up nutrients, and so essentially these submerged aquatic plant beds can be seen as kind of a kidney in the Chesapeake Bay. So because of all of these things, submerged aquatic plants, even though we don't really see them, you really have to get up close and personal, like on a kayak and a canoe, they're kind of hidden out there. They really pack a punch to the health of estuaries, lakes, and streams. And it's really important for us to understand that. So the outline of my talk today is that I'm going to give you a quick tour of representative species, because I didn't want to assume that everybody in this room actually knew anything about submerged aquatic plants. I'm sure some of you do, but maybe some of you, this is really your first introduction. Then I'm going to tell you about the trends in abundance that we see in the Chesapeake Bay. This gets into Bay Optimism. Then we're going to actually have a discussion about the factors in recovery. So I'm actually going to open it up to all of you. I'm going to stop talking, and hopefully you'll have some ideas. And then I'm going to talk about restoration and resilience, some of my work when it comes to the research that I do. So the Chesapeake Bay is actually quite highly diverse when it comes to submerged aquatic plants. There are about 20-some species in the bay, and it's really kind of interesting, the diversity gradient. Dennis was just telling you that I'm interested in biodiversity, right? and in understanding just patterns of biodiversity. And what's really interesting for the Bay is that the low salinity areas, which are in red, so that would be up here, Susquehanna Flats, up here, Potomac River, so the upper reaches of rivers, you have a high diversity of submerged aquatic plants. And as you add more salinity, you actually, species drop out and other species come in. So what I'm going to do here is I'm going to not talk about all of these species. We would be here for a couple hours. But I'm only going to show you representative species, two from the low salinity area, Valsinaria americana, which is the species that I actually have been working on for the last 10 years. And then I'm going to contrast that with Hydrilla verticillata, which is an invasive species. Valsinaria up here is a native species. And then one abundant species in the medium salinity area, which is Rupia maritima, and then the high salinity species, Ostera marina, which probably most people are familiar with. If you are familiar with submerged aquatic plants, you're probably familiar mostly with Ostera. So let me tell you a little bit about my own study species, the wild celery, Valsinaria americana. So it's primarily freshwater, but it can withstand some brackish, some salinity. And so what you have here is that you have some incidences, some observed populations up in the Susquehanna, sassafras, elk, and then coming down into the Bush River, Gunpowder River, Patapsco, also in Patuxent and in the Potomac, kind of where Washington, D.C. is. Where it's absent is actually the Severn and the South Rivers, and we actually looked there just last summer, and we were so hoping to find Valsinaria. I'm going to tell you a little bit about this later, but I'm very interested in understanding whether Valsinaria is locally adapted to salinity regimes. And so I'm really actually looking for Valsinaria populations that occur in higher salinity areas. So I'm actually particularly interested in the Severn River. I'm doubtful we're going to find it in the South River, but the Severn River, it could happen. So Valsinaria is interesting because it does prefer silty and sandy soils, although it can handle muddy soils just fine, or sediments just fine. And in fact, actually, it tolerates high nutrient conditions extremely well. So it does well in eutrophic waters. It also withstands wave action quite well, and it's a superb waterfowl food because just like with sago pondweed that I just showed you, where I showed you the tubers and the esophagus, Valsinaria produces the same sort of underground structures, actually called turions, and so they're a really important waterfowl food in the winter. So this is a really important species and is actually used quite a bit in restoration activities, especially in the freshwater portions of the Chesapeake Bay. Hydrilla now is an invasive species that also occurs pretty much where Valsinaria occurs, except for it doesn't really tolerate a salinity quite as well as Valsinaria does. And so it doesn't go down quite as far as Valsinaria. But still, it occurs pretty much in the same areas, and so you actually see them growing together quite a bit. It is invasive because it grows extremely fast. It actually has a higher water content, and so because of that, it can, when it puts energy into its tissues, it just makes a lot of it, just because of all that water content. It can grow to 7 meters in depth, so it's highly light tolerant or turbidity. Its turbidity tolerance is pretty high. It's also highly polymorphic, which means that depending on the environment, it can produce different sorts of morphologies to withstand different environmental conditions. Now what's interesting, even though this is an invasive species, so right away we're all like, blah, invasive species, no, no, no. It's actually really good fish habitat. Actually, bass fishermen really like it. And the other thing is that it produces such thick mats that actually it's also very good at decreasing turbidity, picking up nutrients and those sorts of things. So even though we all would much rather like native species, this species actually does contribute to the functioning of ecosystems to some extent. Now, I should point out actually is that it has this last point here. It is sort of a boom and bust plant, and actually a lot of submersive aquatic plants have that, where they will start growing in an area and will just boom, produce lots of biomass and expand really rapidly. And then somehow the population goes bust. And so you get these cycles of booming, doing very well, and then crashing, and then recovering again and crashing again. And that happens quite a bit. So let me just show you what I observed with hydrilla at Otter Point Creek, so up in the Bush River area. So this was in summer 2002. This was vegetation density that I observed. And so what we have here is the red areas, you have higher density, and then blue area, you have nothing, okay? So this was in summer 2002, and I went out for basically 6 summers. And so you see a time series here. So this is what you just saw, and then 2003, 4, 5, and 6. And so what you can essentially see is that 2003 it expanded. 2004, you thought that you could just walk on top of the water. It was so dense. And in fact, actually, that picture that I showed you before on the hydrilla slide, that actually came from the summer of 2004. But then in 2005, and especially in 2006, it disappeared, or not quite disappeared, but it went bust. And so there's some really interesting hypotheses that you can now establish to explain why you have these boom and bust cycles. Maybe there's something going on with a plant gets just so productive and so produces all this organic matter, and then that, when this plant senesces and you get all this organic matter going into the sediment, that actually can create some toxicity to this plant, and so therefore the plant goes bust. There's a bunch of different things that I would like to explore here, but I'm not going to get into that. But these boom and bust cycles are really kind of interesting, I feel. So now going into the medium salinity area, one particular plant is Rupia maritima, and it's also a boom and bust plant. It is highly sensitive to wave action, and so if you have a particularly high energy year, let's say a hurricane comes through right when the species is out there, then that can produce a lot of damage to beds. But on the other hand, if the environment is conducive to growth, this plant will take off. So again, you have these boom and bust cycles. Very valuable food for waterfowl. And then finally, we have Zostera marina, or eelgrass. So if you've paid attention to these pictures, they all sort of look alike. They all have these ribbon-like leaves, kind of narrow leaves, and it's so that they can just kind of go with a flow, with wave action, with the flow of the water. So Zostera marina really likes it salty. In fact, actually doesn't really like fresh too much. And so actually maybe that's why we don't see Vallisneria in the southern and the south, because it's really just too salty, and actually Zostera will sometimes grow there, although I think it probably gets a little too fresh there sometimes. So it likes to grow in these saltier areas. High waves are damaging, but it's also a highly valuable food for waterfowl. And so when you put all of these distributions together, so the distribution for Zostera, the distribution for Rupia, and for all the freshwater species, you end up with a total distribution. And this is actually the distribution that we think happened historically. So Orth and Moore in 1983 published a paper in Science that showed this distribution. And so what you can see here is that submerged aquatic plants across all species actually covered quite a bit of the Chesapeake Bay. Well, this isn't the case anymore. And the reason for that is that whereas the watershed might be quite forested historically, because of deforestation, urbanization, as well as agriculture, you end up with a lot of sediment inputs into the bay, as well as you have a lot of nutrient inputs that creates algal blooms and more turbidity, and so a low light environment. And so essentially, if you have high turbidity and high nutrients, you end up stressing the plants. The plants need light, obviously, and nutrients can also be toxic to the plants. And so over the years, submerged aquatic plant beds dwindled. And then something happened in 1972. Do you remember? Hurricane Agnes, right? So this was actually Court Stevenson in 1979 published this paper. So in 1971, we had all these black dots is where we have some observations for submerged aquatic plants. And so at this point already, submerged aquatic plants were stressed by all this pollution that had been happening. And so this was already, these beds were already stressed out. But then look at what happened. So then we had 1972 Hurricane Agnes happening. And then we have, so this is what you just saw. This is 1973 and 1975. And I don't know whether you can see quite that well. I'm going to show you some other ways of looking at it. But what you can tell, for example, look at this. Susquehanna Flats in 1971 was chock full of submerged aquatic plants and empty here. And it seems like it's coming back here, but it's still, the density of points isn't nearly as good as in 1971. So it's basically a perfect storm. We have submerged aquatic plants that are already stressed because of pollution, because of low light conditions in the Chesapeake Bay when it used to be pretty clear. And then on top of that, you have the storm that knocks out a bunch of beds, a bunch of plants, and so now you have very little left. So as I said, I was going to show you, I'm going to show you a couple different slides that show you sort of the trends in submerged aquatic plants over time. So this is data from the Virginia Institute of Marine Science. They fly the bay every year and then delineate where the plant beds are to figure out how much of the Chesapeake Bay is covered by submerged aquatic plants. It's an incredible service that they provide, and it really needs to happen every year just because submerged aquatic plants just come in and out so fast, and they really respond to environmental conditions of a particular year. And so what we have here is in 1978, we have around, what does that look like? Maybe 17,000 hectares, total hectares of submerged aquatic plants. And then each bar here is another year, okay? So what's really interesting then, and this is where Bay Optimism comes in, is that in 2015, we just had a really, really good year. Actually, the best year so far, although some actually get pretty close. The other really interesting thing is, and this is why I was telling you about boom and bust, is that there are fluctuations, okay? So even if so we don't have the data for 2016 yet. I can't show you that. I do know it's high up there too. But if in future years there is a dip, it's not a total cause for alarm because you do see these variations. That's just normal. But what is interesting is that you do have these sort of boom and bust cycles, especially here. It goes whoop down and then goes back up. And so Cassie Gerbitz, who was a graduate student at Horn Point Lab, one of the sister labs to the lab that I'm at, which is the Appalachian Lab, she and her advisor, Michael Kemp, they actually went to the Susquehanna Flats. So this is only Susquehanna Flats data. And so they show, again, this time series going from looks like 84, so low density, and then it seems to recover, and then Hurricane Isabel hits. And then Hurricane Ivan hits. And so you have this dip, but then ever since then, it's really been increasing. So again, there's some Bay Optimism here. It seems like SAV stands for submerged aquatic vegetation. So if I say that, you now know what that means. So things are really starting to look up, but what you need to understand is that when you have these storms happening, that you will probably see a dip again. And so what I want to do here now is just to drive it home. This is supposed to, yeah, this is a movie. So I'm just going to get started. It starts in 1971 and then goes 1974 and kind of cycles through. This is going to take a while. But, you know, just look at it for a while and you can see beds blinking in and out, in and out. But over time, you actually see an increase. There's a general tendency for SAV to increase over time. So we're now starting to get into the 90s. One thing to really pay attention to is the Susquehanna Flats area up here. You're not going to see too much in the South River, so you might not want to look there necessarily. Okay, so we're getting closer here. But if you remember 1971, there are hardly any black spots, and now, you know, we're seeing some blacker areas here. Yeah, see the Susquehanna Flats here, there was one nice big year. Let's see, it expands and expands. There we go, woo! See, it's doing really well. Now what happens when those tropical storms? 2011, remember tropical storms? That was a year. So it contracted again, but then I think 2014, it was the last year. So now what we have, this movie ends with this colorful map, and that is where all of these different distributions were all put together in one map, this map here. And when you see the dark areas, it's actually dark blue, that means that you had submerged aquatic plant cover for at least, I'm trying to recall here, but 10 years or so, okay? And the hotter the colors get, the less often you saw submerged aquatic plants there, okay? So essentially where you have these dark areas, that means that these are sort of these are really good beds. They're really stable beds, and the hotter colors means is that they just kind of go in and out much more. And so if we actually want to look at the Susquehanna Flats, because this is just such an interesting story with, remember in 1973, the Stevenson et al. Plot, all of the plants got wiped out. And this is, you know, Susquehanna River comes right in here. So it's really just a fantastic kidney if it could work that way, right? Because it could really allow sediments to settle out, pick up nutrients and things like that. And so what you have here is a bed that actually stays around actually pretty much every year. And then it expands and contracts depending on different environmental conditions. And so then what one of my colleagues does at the University of Maryland Center for Environmental Science is he puts out these, this is Bill Dennison, he puts out these report cards. You might have already seen that, some of you have. And so they're really interesting in that they're kind of tractable. You know, we've all seen report cards before, right? And so, so in 2006 here, this is 2006, he gives, or his whole group gives each tributary essentially a grade. And I'm not going to get into the methods here. But in 2006, the score was 37%. So I don't know what that means when it comes to what grade that is, but it's not a very good grade at all. Oh no, it's a D plus right there. D plus, not so good. In 2015, it at least got a C, and you might not actually be able to see that all that well, but next to the grade, there is this box here, and if it's green, that means that it's improving, okay? And then this line means that, it means that it's improving. And so essentially, all of these areas up here in the north, they're improving, as well as some of these areas down here. So this is quite interesting. And the reason for that is that there's 3 indicators of bay health that score extremely high. There's chlorophyll A, water clarity, and look at this, aquatic grasses. So they're doing really, really well, and they did really well in 2015, scored really high, and then that improved just the health of these different tributaries. So aquatic grasses really pack a punch here for estuarine health. Now, so this all is wonderful, but I'm a scientist, and I always have to look at things critically and look at them in different contexts. So this is this graph that you've already seen, but now the y-axis is a little bit different. And the reason why it's different is that I put up here the restoration goal. So this is a restoration goal that EPA has put forth, saying that we would like to restore the Chesapeake Bay to 75,000 hectares total. Okay, so once we put it into that context, yes, we see improvements, but we still have a way to go. You know, we can't just all go, we made it, you know, SAV is improving, now we can go on to tackling other environmental problems. Actually, we still got a ways to go. And we can look at it even in a different light. Are you ready? Mm-hmm. If we put historic cover up here. Oh, maybe I shouldn't have shown you that one. Okay, so that means we still got a ways to go, right? I think we can all feel good that our estuary is going in the right direction, that submerged aquatic plants do seem like they're coming back, but we still have ways to go when it comes to restoration. And so this is where I said that I would try and get you involved. So what do you think are some factors in the recovery? What can we actually do individually or as a community to help these submerged aquatic plants coming back? Yes?
Well, I think slowing down development in communities next to the bay is probably the most important thing.
You're right, yeah. Yeah, the Chesapeake Bay is just growing. I don't know right now what the trends are, but there's more people moving in. And so there's just a lot of pressure on the bay just from that itself. Absolutely. Yeah.
Encouraging living shorelines.
Living shorelines. Okay, can you expand on that? What do you mean by that?
Well, I know that certain individuals or groups of individuals are actually going in and doing the restoration of the plantings off of their own properties.
Mm-hmm.
So I know that that's a beginning effort of trying to raise awareness and have individuals take some responsibility for whatever, if they have shoreline properties.
Mm-hmm. And then living shorelines would reduce erosion, reduce nutrient runoff.
Right, create habitat.
Yeah. Okay. Yeah, back there.
Stream restoration and stormwater management.
Okay. Yeah. Yeah, that's good. That is something that's near and dear to my heart because I'm a wetland ecologist, and so it's especially so stream restoration I think can also include wetland restoration, like along streams, and floodplain restoration. The thing is that wetlands are essentially like a sponge, and so if you have a storm, rather than the storm just running off right into the stream and then gushing out into the estuary, taking with it all these sediments and nutrients, the wetlands can actually pick all that up like a sponge and transform the nutrients and basically keep it in the system. So yeah, stream restoration, stormwater abatement, all that is really important. Yes?
When you have these plants absorbing these chemicals and pollutants, when the plants die, what happens to them?
Yeah, so whatever is located above ground as the tissue decomposes, I guess that could be released back into the water. But often when plants senesce, they'll first actually translocate the nutrients into the root systems to kind of preserve their nutrient balance a little bit, and so that they have that for next year's growth. And then also some of these some of that biomass can also be worked into the sediment, so then it stays in the sediment. But you're right, as the plant senesces, some will go back into the water. But I think it's a net decrease, like more is brought into the system and into the sediments than is released back out into the system. Yeah.
How about cleaning out behind Conowingo Dam to make space for more sediment there?
Yeah, that's a big one, isn't it? There are so many nutrients in that sediment. It's crazy, and that continues to produce it's a pollution source essentially. And so the best thing is to actually get all that out. But that's a tough one. And scientists are actually looking into that. It's a good one, yeah. Yeah, back there.
What impact does fertilizer on farms and also lawns by the water have on submerged aquatic grasses?
Big, right? Totally. Because if you don't use fertilizer wisely, that all then, rather than getting picked up by the plants and getting immobilized and staying in the system, just gets washed into the streams. Yeah. Yes.
Can you do assisted migration, like assisted planting? From
Yeah.
poor areas to other places?
Yeah, so you're thinking more like climate change, and so the species that we have here might not be as well adapted and can't interact with the environment as much as maybe some other plants can. It's definitely something that people are thinking about at this point. Yeah, go ahead. And then back.
To the local level, out on 4-acre tidal pond, and we've had grasses coming back the last couple of years. Don't know if they're invasive or not, but some of the residents don't like them because it bothers the boats. And so they put chemicals in there. And I'm trying to tell them that these chemicals aren't good. Am I telling them the right thing? And what do these chemicals do?
Yeah. So herbicide use it can be an issue, especially herbicides that are not biodegradable and that then stay in the system and could be in the system for a long time at a very low level, but these plants are still exposed to it chronically, and so that could stress the plants and could produce an impact. I think it can have an impact for sure. Yeah.
If memory serves me enough, I live on Herring Bay, on the water, and your vegetation coverage chart, year by year, almost exactly mirrors the number of trumpet swans that winter in front of my house every year. And I moved there in '95, over 200. And it went down when we had the hurricane, it went up a little, and it came down again. And now this year, for the first time, we're over 100 again. We're up to 120 this year.
Wow.
And
Okay.
they come at Thanksgiving and they leave at Easter.
Wow. Yeah, that's great. That's awesome. Yeah, so yeah, they're totally, if you don't have anything in the sediments for them to feed on, that they won't stay, obviously, so we need to bring them back. Okay, are you ready now for my answers? I think you pretty much got them. I think you got them all. So we need to manage sediments and nutrients coming off of land, so living shorelines are really important. Managing our farmland so that there's something growing on this farmland rather than, you got storms that then wash everything into the streams. I don't think you guys picked up on this. Boating, fishing, and aquaculture practices. This is actually not from here. I just think this is such a pretty photo. I had to show it to you. It's from Florida. But the reason why I'm showing it to you is these are all boat scars, okay? So boating can really scar beds. Then yay for herbicide use, or not yay, but good for you to have said that. Storm abatement, and then assisted migration gets at this last point, which is genetic diversity, which is what I would like to talk to you more about. So I think you hit really on the big ones. There are many factors that lead to the recovery, and there are some things that we can do individually. We all can be empowered to do something that would decrease nutrient runoff, for example, not using fertilizer on our lawns, not boating in SAV beds with propellers that could scar them, those sorts of things. And some things we can maybe do as communities as well. Yeah.
See any effect from sea level rise?
Is this Dick?
Yeah.
Yay! Not me personally, not yet. But with sea level rise, you have more salinity intrusion that then affects some species more than others. But there could be effects from climate change, from sea level rise. The same thing also with temperature. One of my colleagues has shown that the temperature of the bay is increasing in some spots more than others, but overall it's increasing. And that has an effect on some species that might already be sort of at their northern limits.
Before you go on, could you explain the storm abatement? I mean, what can we as individuals do to address that?
Yeah, so for storm abatement, I was thinking more of reducing runoff from storms. So keeping stormwater close to us rather than it just all washing downstream through the watershed into the estuary. And so one thing that we can do, rain barrels. As communities, we can try and restore wetlands. So then let's talk about restoration a little bit. Restoration, the ultimate goal is to reestablish self-sustaining ecosystems that will be resilient to future perturbation without ongoing human input. So I would like to underline self-sustaining and resilient. Okay, so this is important. We can put plants out there, but they might not actually be resilient. We need to put thought into what it means for population to be resilient and self-sustaining over the long term, so that we have them year after year after year. Yeah.
Does that include introducing alien species into the environment?
No, I would never ever suggest that. Yeah,
Hydrilla seems to be working well.
Hydrilla seems to be working well, but I would never suggest actually introducing invasive species because there's actually native species that can do the job as well. So we should focus on the native species first. Like Valsinaria can do the job just as well as Hydrilla can, or Elodea, which actually looks very similar to Hydrilla. Yeah. So there are different ways to get at resilience. So one of them is that species can actually acclimate to different environmental conditions. And so this is what's called phenotypic plasticity, which submerged aquatic plants actually have quite a bit. They're phenotypically very plastic, which means that they can kind of change their morphology so that they're more in tune with the environment. They can also adapt to the environment, and this is where we get into genetic variation and also associated phenotypic variation. So just like all of us look a little different, right? Our genetics are different, and so we have different noses, different eyes. We are just very different. And so we are phenotypically, there's phenotypic variation in this room, and there's also genetic variation in this room. Same thing goes with populations, with submerged aquatic plant populations. We can also relocate different species so that they are more in tune with the environment, just like what this gentleman said with assisted migration. Okay, so that's what this is here, assisted migration. So there could be natural dispersal, local restoration, or assisted migration where we actually introduce a species from somewhere else into a new area. So that kind of gets into maybe not necessarily invasive species, but sort of if a species can't actually disperse itself into a new area, we might consider actually moving it and seeing whether it can grow. This is kind of contentious, and some people would say, no, this is actually not something that we should do, but people are researching that right now. And then the other thing that could happen with plants, if they can't acclimate, if they can't adapt, or if they can't relocate, is to become extirpated. So those are our 4 options. So looking at genetic variation or considerations really quickly, since I can't assume that all of you know something about genetics. So genetic considerations for resilience and restoration are that we can look at different genotypes or a number of alleles within a population, which contributes to growth and survival, resistance to disturbance, as well as potential for reproduction. I'm actually going to show you some data on that. Then we can also, when it comes to genetic considerations, we can look at levels of heterozygosity, where low levels of heterozygosity often means that an individual is less fit and cannot grow and reproduce as well. And then we can also look at adaptation of individuals to the local environment. Okay, so we have number of genotypes, we have heterozygosity, and we have local adaptation. There's many different ways we can look at genetic variation. And so we looked at this in the lab, in the field, in the greenhouse. We collected leaf tissue from ramets. Those are submerged aquatic plants. Many of them are clonal, and my study species, Vallisneria, is clonal. So each shoot that it produces is actually a ramet that can still be connected. So we collected leaf tissue from these different plants, as well as whole plants that we grew in the greenhouse. And then we conducted experiments on genotyped individuals. We collected samples from Susquehanna Flats area, Sassafras River, Bush River, Gunpowder, those areas here, and then throughout the Potomac River, actually, where Vallisneria occurs. And so what this actually shows here is the different bubbles shows genotypic diversity, so how many genotypes are in a population. We sampled 30 individuals per population. And so the bigger the bubble is, the more genotypes there are in the population. So you can see up here it's actually quite genetically diverse, but then other areas are less so. But actually the take-home message for the Chesapeake Bay is that actually we have, the Vallisneria populations are actually genetically quite diverse. It's actually a really feel-good story. Another way of looking at Bay Optimism is that genetic diversity of Vallisneria is looking really good, which means that it should be able to adapt to different environmental conditions. So there are a couple sites, though, that heterozygosity was actually pretty low in the Potomac River. And then when we actually look at how different the populations are genetically, we can look at what's called genetic differentiation. This is a program called Structure that we can use here. And so when we have different colors here, that means that the model, that this genetic model is saying that these populations are different from the ones in the red, from the ones in the green. But something to look at here is that the green and yellow area, there's a lot of what's called admixture, where the individuals could be from one or the other. There's a lot of mixing going on here. And so from this output of the model, we can then say that the populations up here in the Susquehanna Flats are genetically different from the ones here in the Central Bay. And then the lower Potomac River and the upper Potomac River are also genetically different, but there's some more mixing going on with that river system. So this is kind of interesting, and it suggests that we need to actually be careful when we restore populations of Vallisneria in the Bay, which we do quite a bit. School groups help. The Department of Natural Resources does many restoration projects. Is that we don't necessarily, or we need to be careful about if we take individuals from, let's say, the upper Potomac and we plant them up here in this area. We may not necessarily want to do that because the genetics up here might produce a different tolerance to environmental conditions, may produce a different phenotype than what we have up here. And so restorations should actually, to do it right, we should actually stay within these different regions. And the Department of Natural Resources has really heard us and is actually using using this approach now. They actually ask us, is it okay to use these plants up in this area? And we can then say, well, it's actually coming from a different area, or yeah, go ahead, it's from the same subregion. Yeah, go ahead.
Is there such a thing as aerial seeding with these types of plants?
An area of seeding?
Well, I mean, like the birds eat seeds and then they drop the seeds and things grow in other places. Is there some sort of seed that comes from some of these species that you could use to increase the distribution?
Yeah, so you can actually, you can harvest the seeds. They produce seed pods, you can harvest the seeds. And so, but the thing is that if you harvest seeds from, let's say, the upper Potomac River, you'd want to use those seeds to restore populations in the upper Potomac River, not necessarily within any of the other regions, just because there might be some local adaptation going on in these different regions. So another way of looking at this genetic diversity is to put it again in two different contexts. Remember, I showed you the patterns of abundance and how you can compare that to, well, this is our restoration goal and this is the historical abundance. Well, we can do the same thing with this data. So we can compare the Potomac River to the Hudson River to the Kennebec River, which a PhD student has done at College Park. So here we have the Potomac River. We have genotypic diversity with these different bubbles where hotter colors are higher genotypic diversity, the Hudson River and the Kennebec River. And what you can see is that the Potomac River is pretty much on par with the Kennebec River, but the Hudson River is actually pretty depauperate. And it turns out that when the storms of 2011 came through, the Hudson River Valleson area really was decimated and is just now coming back. It's taken a long time to come back from storms. We can also actually look at the literature from throughout the world when it comes to genetic diversity within any submerged aquatic plant. And then, so that's these gray dots. So that's kind of our background. And then we can actually put our data on top of it. And so we have the species mean, we have the population minimum, population mean, and max. And what we can pretty much see is that for most of these, we're right in the middle of the genetic diversity of any submerged aquatic plant worldwide. So the Chesapeake Bay plants are actually in pretty good shape. Same thing for allelic diversity. And then we can do experiments in the greenhouse. Lots and lots of experiments. We did that for two populations. We focused on Piscataway Park from the Potomac River and Hawks Cove, which is just below Baltimore. What we did was we used two different soils. This is actually the Piscataway soil, and this is the Hawks Cove soil. And then we put the different population into each other, so we have an away versus home sort of experiment. So populations were grown in their own home soil and also in the away soil. On the x-axis here, we have different genotypes. And then on the y-axis, we have the number of leaves. And what I did is I ranked, basically, the number of leaves is essentially productivity of different genotypes. So I ranked the productivity of these different genotypes. And what you can see is that they all differ. They're not all the same. You know, they might all look the same when you just look at different genotypes. You think, oh, they're going to do the same thing. But actually, they're very different. And then, for example, when you look at the number of roots, we did all kinds of measurements on these. Same ranking. What you can see here is that the most productive genotype above ground actually did well, but another one didn't do so well at all. And it turns out that every genotype does its own thing. They essentially all have, to some extent, their own personality, their own role to play in the environment. They're not all the same. So here what we have is, on the x-axis, we have proportion of heterozygous loci. Essentially, that means that as you increase on the x-axis, you get more higher heterozygosity, which is a good thing. On the y-axis, we have tuber weight up here and leaf area down here. And essentially, the take-home message here is that as you increase heterozygosity, you actually increase plant performance. Okay? But it's a little bit confounded by that Piscataway plants actually overall were lower in heterozygosity than the Hawks Cove plants. But overall, if you have higher heterozygosity, you actually increase performance in plants. It's something that you would expect from the literature, but that I didn't expect to find in my own data. Yes?
Was there any significant difference in the nutrients available in the soil between the two places?
Yes. Yeah, there definitely was. Hawks Cove was much richer, much more conducive to growth than Piscataway soil is. Yeah. One thing that was really interesting is that when we look at the number of genotypes in a population, so we planted 2 versus 4 versus 8 genotypes in little buckets in the greenhouse, what we saw is that this is number of flowering events, that the more genotypes were in a population, the more flowering events occurred, which means that there was more opportunity for populations to mate, or for individuals to mate and to breed. These are, the Valsinaria is a dioecious plant, which means there's males and females, and you need both for them to combine and pollinate, for the male to pollinate the female and then produce seeds. So that was really interesting. Another way of looking at that is again, we have genotypic diversity on the x-axis, and on the y-axis, we have number of experimental populations where we had both sexes in the population, females only, male only, and absent. And what you can see here is that, just look at the black bars. As you increase genotypic diversity, you increase the chances of having both sexes in the buckets, which means that they could mate and produce seeds, which is a good thing. And then at the same time, if you look at the absent histograms, that goes down. And so essentially, again, that means that more genotypes yield a higher probability of having both males and females and a lower probability of non-flowering individuals. Okay, so more genotypes, this is essentially my summary here. More genotypes yields better performance. Individual genotypes and population sources affect ecological performance more than individual heterozygosity. So it really depends on what genotypes you actually plant in the population. And then there was no strong, consistent evidence of benefits of genetic rescue. I actually didn't really show you this data, but you were just asking about whether the different sediments were different, whether there was different nutrients, and one was richer than the other. And it turns out that when you put any of the individuals into that richer soil, they just did better. But what was also interesting is that in the less rich soil, you actually had more opportunities for selection happening for that population that came from that soil. Like it actually did better in its home soil. It's actually kind of hard to explain without the slide. But essentially, we saw some evidence of local adaptation, but it wasn't where the population in its native soil always did better than in the away soil, and it's because of the difference in sediments and one being richer. And yeah, so some evidence for local adaptation that needs to be confirmed. And then finally, restoration practices that consider local adaptation of Chesapeake Bay, Valsinaria americana populations are advised. And the reason for that is that we have these different genetic structures going on in the Chesapeake Bay, which could mean that there's some local adaptation going on, but we still need to research that a little bit more. And with that, I just need to acknowledge a bunch of different students, and especially my collaborator, I've collaborated with her for the last 10 years, and she's just fantastic to work with and incredibly smart. And with that, I'll take any more questions. Thank you.